Somebody confirm you can hear me?
Yes.
Thank you. Guess we should get chapter 3 up too. All right, before we begin, are there any questions?
I just have a question about next week's test.
Uh-huh.
What would the material be covered on that test, since we just had one this week? Like, what should we study?
Just a minute. Why is that saying open? Did not Zoom open. All right, let me close that down. It's on the schedule of the syllabus. Let me show you that in a minute. I've got to figure out how to get to my Zoom. Share my screen here. All right, you'll be seeing my screen. Come to the syllabus, and for quiz 2, it will cover labs 3 and 4, chapter 3, and the end of chapter 2.
Oh, gotcha. That makes sense. Thank you. Can I just ask, when you say the end of chapter two, do you have specific page numbers that we should be focusing on for the end of chapter two?
It's about where we left off the first week. Um, I could give you a specific page number, but off the top of my head, I can't. So, what you can do is email me that question. I'll try and get that to you.
Okay.
All right, any question? We're supposed to be on chapter 3. We're not there yet. We need to finish up chapter 2. There will be a lab tonight, lab 4 on stains. Any other questions before we get started? Come on, let's go. All right, let's go to chapter 2. I have that we had discussed RNA last time. Let's talk a little bit about ATP, adenosine triphosphate. ATP is the primary energy source for all cellular activities. Whenever the cell needs energy, it usually gets that energy from ATP. ATP has ribose, that's the sugar right there. Same sugar in RNA, by the way. Three phosphate groups attached to it, and then adenine, one of the nucleotide bases in DNA, but obviously this is ATP and a energy source. It's an energy source because there are two phosphate bonds with the squiggly line, that one there and that one there, and they're high energy bonds. When the cell wants energy, it'll just break off that first phosphate group, and that will release a large amount of energy. But some of that energy can be used for the cell's energy needs. If the cell needs more energy, it can even break off that second phosphate group to release more energy. Any question about any of that? ATP is made by dehydration synthesis. What you do is you get adenosine diphosphate, that's ADP, adenosine 2 phosphates, add it with an inorganic phosphate, add energy in, and then you get water and ATP, a dehydration synthesis. You can also break ATP by adding water, and that will take off this phosphate group, making ADP or adenosine diphosphate, and that will release a lot of energy. Much of that energy, not all of it, but a lot of that energy can be used for the cell's energy needs. Any question about any of that? So where do we find ATP? Anyone guess? In cells and tissues that if the tissue needs energy. All right, last slide. Hydrogen bonds. What molecules have we discussed have hydrogen bonds that give the molecule important properties?
Water.
Water, yes. What about another molecule? There's one you should remember.
DNA.
Yeah, DNA. It has hydrogen bonds holding two strands of DNA together. Actually, sugars and carbohydrates also have hydrogen bonds. We'll not really talk much about those, but that's why sugar dissolves in water. What would happen to these molecules if all the hydrogen bonds were to disappear in the universe? Well, would you have liquid water?
No, we'd probably have gas.
Yeah, you wouldn't have liquid water. So you would have gaseous water, and then you wouldn't have double strand DNA as well. All right. Any questions for chapter 2? If not, let's close it and begin chapter 3. Here you're seeing the major goals and the rough outline of chapter 3, observing microorganisms. Know the terms, what's resolution, refraction, what's a stain. There are basic and acidic stains or dyes, and dyes and stains are used synonymously. What is a smear preparation? What is a mordant? What is peptidoglycan? And then 2, understand the sizes of organisms and the properties of microscopes. Understand the major differences between a light microscope and an electron microscope. And then 3, understand why staining improves resolution. And know there are 3 types of stains: a simple stain, a differential stain, and a special stain. Understand the gram stain, its procedure, and the basis of its differential staining. Any question about this slide, major goals and the rough outline? All right, let me blow that slide up. So when we're observing microorganisms, you should know that we will use measurements of the metric system. And you need to know the metric system for quiz questions, as well as the fact that science and medicine use the metric system. Any question about any of that? All right, you should know that 1 millimeter equals 1,000, 1 meter equals 1,000 millimeters. And you probably should know that 1 millimeter equals 1,000 micrometers. A micrometer is let's see, is that a millionth? 1 times 10 to the negative 6 of a meter. You should also know a nanometer. That's 1 times 10 to the negative ninth, or a billionth of a meter. Let me blow this up just a little bit. So you should know a kilometer. That is 1,000 meters. The meter is the basic unit of the metric system when measuring length. You don't need to know a decimeter, that's 1/10 of a meter. You do need to know a centimeter, that's 1/100th of a meter. And then a millimeter, that's 1/1000th of a meter. A micrometer, that's 1 millionth, or 1/1000 meters, meaning 10 to the negative 6 meters. You should know a nanometer because that's the size we use when we're using an electron microscope. You don't need to know a picometer. You should know that the micrometer is the size that we see under the light microscope. Now, admittedly, a micrometer is under high power, but you can see down to a micrometer and a little smaller than that under the light microscope. Not a micrometer, yeah. Also call it micron, but we'll use the term micrometer. So you should know the prefixes, what they mean for the ones I mentioned. And then when you're talking about a gram, that's the basic unit of weight and mass. you use these same units. There's a kilogram, a centigram, a milligram. So you should know most of the metric equivalents. You don't need to know, like I said, the picometer, and you don't need to know a decimeter either, but you should know these others. And usually when I'm working in metric, I will simply take whatever I'm doing. Like if I'm asking the question, oh, I've got a large number of nanometers and I have to convert it into, let's say, centimeters. I usually convert the nanometers to meters and then convert the meters to centimeters because I know that there are a billion nanometers to make 1 meter. And I also know that there's 100 centimeters to make 1 meter. But I don't know how many nanometers directly make a centimeter. So I just usually convert to a meter and then convert to a centimeter. And that's just an easy way to use metric. If you want to take um, the entire metric system, you can remember that it's based on a unit of 10. So a meter is one. A decimeter is 1/10 of a meter. A centimeter, 1/100th of a meter. A millimeter, 1/1000th of a meter. You do not need to know the US equivalent. We will not convert between US units and metric units, but you do need to convert between metric units. Any question about any of that? I think in the lab I've got, let me see if I can go to the lab page here. I'll show it in the lab, but I've got some practice metric quizzes on the lab here. So let's go to that. There we are. Nope, it's actually the practice quizzes are in the lecture. So there's 3 practice quizzes. Realize when you click on this link, it's not going to open in Canvas. So it's going to do this and tell you, oh, that did open in Canvas. That's different. Let me try another one here. Normally it doesn't open in Canvas because it's not a page in Canvas. There, that one it can't open in Canvas. I don't know where the first one is opening. It used to never. But if you click on this link here, it'll take you outside of Canvas and then take you to the quiz. Test your knowledge of meters, grams, and liters. Start now. And then it's saying long distances are measured in which one here? Meters, kilometers, centimeters, millimeters. Kilometers would be the largest. We're going to take kilometers because it's large distances. And it tells you if it's green, it's correct. Good job, feedback. The metric base units include, we're going to get a wrong answer here to show you what it looks like. We're going to say liters, yard, pound, which is obviously wrong because that's the English system. And submit answer. It'll say red, your response, feedback, sorry, try again. And the correct answer, oh, I'm going to start the quiz over again. Well, the correct answer is meter, liter, and gram. Oh, it tells you right there. Never mind. Correct answer. Any question on how to use that quiz? Um, this first one isn't going to work well in Canvas. Yeah, so you can barely even see it. So let's go ahead and go outside Canvas. And there it is there. Select the base unit of the metric system for length, a meter. It's green, so that's the correct answer. Keep going, bullseye for question 1. What's the first name or nickname you'd like us to use? Well, you don't need to do that one, but let's go on to 3. Select the base unit of the metric system for volume. It's a liter. I don't think you want to go on that. Continue button. Yep, right there it tells you. So just move down. If you hit the continue, it's going to try and download something, so don't hit the continue button. So be careful of the buttons on these pages because this is off of Canvas and you click on it and you'll start to try to download something and you don't really want to do that. All right, any question about any of that? This is the second reason why you should know the metric system. We were looking at all the countries in the world that are shaded in red that do not use the metric system. And two of them are actually slowly converting to using the metric system. The United States is slowly converting to using the metric system. If you notice, sometimes on the speed limits on the road, you'll see it in large print, miles per hour, and then in smaller print, kilometers per hour. And then on most cars, at least most new cars, it'll have in larger print the miles per hour, and then in smaller print the kilometers per hour. We have been slowly converting to using the metric system. I think it was it Andrew Johnson? No, Andrew Jackson, the third president of the United States. Andrew Jackson passed legislation to slowly convert the United States to using the metric system. And we've been very, very slowly going to convert the metric system. I don't remember what the other country is. So there's only one country in the world which is not, or at least 10 years ago, was not converting to using the metric system. You should know the typical size of microorganisms. A eukaryote cell, on average, it's about 10 micrometers to 500 micrometers. It obviously depends on what size you're looking at, cell you're looking at. For a prokaryotic cell, actually the mean size is around 1 to 2 micrometers. A long prokaryotic cell is about 10 micrometers long. So that'd be the one of the biggest. And then the smallest prokaryotic cell that's viable is 0.4 micrometers. The very smallest prokaryotic cell, which they think is not viable, is 0.2 micrometers. So a prokaryotic cell is about 10 times smaller than the average eukaryotic cell. And then you can have a virus, which is about 10 times smaller than the prokaryotic cell. For viruses and the diameter, their size range from 0.02 micrometers to 0.044 micrometers. That would be 20 nanometers to 440 nanometers. So yes, the very largest virus gets up to the size of the smallest prokaryotic cell, and then the very largest prokaryotic cell gets up to the size of the smallest eukaryotic cell. You can take the length of the virus. Ebola is the longest virus, close to about 1 micrometers long. However, Ebola is very skinny, and so its diameter is smaller than the megavirus, which is about the biggest virus. There is one other virus which is bigger than that, but I don't remember the name of it, and it only infects amoebas. So the largest virus, by the way, are the viruses that affect amoebas. You don't need to know that, but just thought I would mention it to you. So when we're looking at things, you should know the size of things. A person, about the biggest thing we'll study. We will obviously briefly mention elephants and whales are the biggest life form currently on the planet. We're not going to study them, but we will study diseases of humans. So about the biggest thing we'll study are humans. And then the chicken egg, that is the biggest cell that you have ever seen, and that is the yolk of a chicken egg. I know you've all seen the yolk of a chicken egg. I even once had a student who'd seen the yolk of a turkey egg, which is even bigger. But most of us have not seen the yolk of a turkey egg. I've never seen the yolk of a turkey egg. The biggest cell on the planet is the yolk of the ostrich egg, and very few people have seen that. The smallest cell you can see essentially is the frog egg. You can see a little bit smaller than that, but frog egg. And then we have our eukaryote cells, which you need a microscope to see, at least most of them. And then we have prokaryotic cells, which are smaller than a eukaryotic cell, about 10 times smaller, most bacteria. Smaller than most bacteria are viruses. Need an electron microscope to see, at least most viruses. I guess the very biggest you might be able to see with a light microscope, but most you would need a electron microscope to see. Smaller than a virus would be a large molecule like a protein or a lipid, very large protein or lipid. Small molecules are smaller than a large molecule. A small molecule would be something like water or carbon dioxide. And then smaller than that, the last thing we'll talk about are atoms. There are things smaller than an atom, like a quark, but we're not going to talk about them. All right, this slide is showing you the basic light microscope. This is a compound microscope, compound light microscope, and that means it has two or more lenses that magnify, the total magnification being the magnification of the objective lens times the magnification of the ocular lens, right there. A typical microscope has a 4x, a 10x, a 40x, and a 100x objective lens. And all of the microscopes in the microbiology lab have these objective lenses. And they all have a 10x ocular lens. As far as I know, every microscope, that's at least a compound microscope, has a ocular lens of 10x at Clark College. You don't have to have 10x. The very best microscopes in the world have 20x. And I have seen some child's microscopes which only have a 4x ocular lens. You should know the parts of the microscope labeled here, although you don't need to know the prism. You don't really need to know the path of the light either. Just need to know the light comes from the lamp, comes through the condenser, shining the light in a cone of light on the specimen, which the light is then collected by the objective lens. You should know that, but the rest you don't need to know about the light sources. You should know about the objective lenses here. I'm not sure. Oh, sorry. I know why there's two, and that's because there's a 4x lens, a 10x lens, a 40x lens, and a 100x lens, 100x being there. That's why they've got pointing two here. There's only one objective lens, because this looks like it's two, but there's only one in use by the microscope at a time. Obviously, there's four different objective lenses. I talked about the condenser lens, showing a cone of light on the specimen, a diaphragm, which opens and shuts like a shutter. We'll take a look at that. And then the lamp or the illuminator and the base of the microscope. You could also learn the arm of the microscope there. You can hold it here and then hold it with the under the base, your second hand. But you should know this part, the body tube or the arm of the microscope. You should know the objective lens, the stage holding the slide, the stage clip, which is shown here but not there's no line pointing to it. The condenser lens here, the iris diaphragm there, and on some microscopes, The condenser is below the iris diaphragm, so don't let that confuse you. On this one, the diaphragm is at the bottom, but at some, the diaphragm is above the condenser. And then the light source. You should know the focusing knobs. There's the coarse focus knob, the big one, and then the fine focus knob, the little one. Let me see what else is on that page. All right, that's it. I could see something there, but I couldn't see what it is. This is a simple light microscope. It is a simple light microscope because it only has one lens that magnifies. So a simple microscope is nothing other than a super duper magnifying lens. This microscope was made by Antony van Leeuwenhoek, and you actually stuck the specimen on the pin there, and then he ground his own lenses. That's why he was the best microscope builder of his day. He was the best lens grinder of his day. The point is, a simple microscope only has one lens. A compound microscope has two lenses that magnify. All right, you should know the resolution of a microscope is the ability of a lens to distinguish two points as separate, independent points. Like when we look here, this is what we're actually looking at, but under the microscope, it's going to look like that. You can make out the two separate independent points so that these points can be resolved as separate independent points, but these points cannot be resolved. They are just a blur of everything together. Any question about any of that? So the resolution is these can be resolved as independent points, and the light microscope, the best resolution you can get is 2 points have to be at least 200 nanometers apart, about that distance here, to be able to resolve them as separate and independent points. So a microscope with the resolving power of 0.2 micrometers can distinguish between 2 points that are separated by more than 2 micrometers. 2.2 micrometers is 200 nanometers. And so 2 points, if we can resolve them, have to be separated by more than 200 nanometers, because that's the minimum a light microscope can resolve. And that resolution limit is the limit of white light. The best white light can resolve is between 200 nanometers apart. And that has to be do with the, excuse me here, someplace I've got some water. The best light microscope can resolve is 2 points separated by 200 nanometers, and that's because that's the limit that white light can resolve. Anything beyond closer than 200 nanometers apart, the wavelength of light will not allow you to resolve the points as two separate independent points. So it has to do with the diameter of white light. And I used to know this. I think it's something like half the wavelength of white light, 200 nanometers. So that means the best compound microscope can only magnify about 2,000 times. That's a 20x ocular and 100x oil immersion lens. That's the best magnification we can get, because if we were to use a higher magnification, you will not get better resolution, because 200 or 2,000 times approaches the resolution limit of white light. And so if you magnify further, it's empty magnification. because you cannot magnify any better than the diameter of the wavelength of white light. Any question about any of that? And it's not exactly the diameter. I think it's half the diameter of the wavelength of white light. But the point is, the resolution limit of white light is 200 nanometers apart, and any points that are closer than 200 nanometers apart cannot be distinguished as two independent points by a light microscope. And these points are about 200 nanometers apart, so we can resolve them. These points are less than 200 nanometers apart, so we cannot resolve them. All right, let's talk a little bit about refraction. Refraction is the bending of light when it goes through a medium and then leaves that medium and enters another medium. So when we're looking at the pencil here, it looks like the pencil is bent right there, and that's because light has a different refractive index in water than in the air, making it look like the light is actually bending right there. And actually, the light is bending right there, but the pencil is not really bending. And this is what the artist's rendition looks like. The pencil looks straight until you hit the water, and then it looks like the pencil bends. This is actually important for life, like the bird's eye will see the fish seen here in the water because of the refraction of light at the interface of the water and the air. The fish is actually down here, but the bird will see the fish here. And the bird instinctively knows when it's trying to catch the fish to put its feet down in this position, even though it sees the fish in this position. Any question about any of that? So, as I stated, light going through glass can bend when the light leaves the glass and then enters another medium, which would be the air, causing some of the light rays to bend at the glass to air interface. If we were to use oil, oil has the same refractive index as glass, so that the light ray hitting the glass slide will go through the glass and then enter the oil, and there will be no bending or refraction of light at the glass to oil interface because oil has the same refractive index as glass. And then when the light goes from the oil to the glass of the objective lens, once again the light will not bend. Whereas when the light goes through the glass of the slide, some of the light rays will bend at the glass air interface, and then some of them won't, but some of them hitting the glass of the objective lens will bend at the air to glass interface. And so the light is actually refracting coming out of the glass slide, entering the air, and then it's going to refract again, leaving the air and entering the glass. So that we can say some of the light rays coming through the air will actually not be bent, will not refract when they leave the glass, enter the air, and then enter the glass of the objective lens. And that's fine. Some of the light rays will be bent, refracted, when they leave the glass and enter the air, and then they'll be bent again when they leave the air and enter the glass. This one will be collected by the lens as well as that one. But this one here, when it is refracted by the glass air interface, it's refracted at such a steep angle that this light ray does not enter the objective lens. And this one here when it's refracted by the glass, it's refracted at such a steep angle, it never enters the air. It's refracted back down into the glass. So these two light rays will not be collected by the objective lens. And if we were to use oil, this one and here will be collected by the objective lens. That one and this one, of course, will also be collected by the objective lens. And that means more of the light rays leaving the glass slide will be collected by the objective lens, making the resolution and clarification of the object better when you're using oil than when you're not using oil and the light is refracting from the glass to the air interface and from the air to the glass interface. So therefore, immersion oil is used with a light microscope, especially with the oil immersion lens, so that more of the light goes into the lens, can be collected by the lens, so there will be greater resolution. Any question about why we use oil, sometimes with a light microscope? Like when you're using the oil immersion lens. All right, this slide is showing you bright field illumination. Bright field illumination is what you normally have in a light microscope, compound microscope, but it doesn't matter. It's a microscope. And that is light leaves the source, the lamp, is collected by the condenser lens, which puts a cone of light on the specimen, and that light goes from the specimen to the objective lens, and then is collected by the objective lens. So bright field illumination is the type of microscope we have in our labs. Compared to dark field illumination has an opaque disc in between the light source and the specimen, so that any light directly coming from the lamp will be blocked by the opaque disc, and that will make the background of the slide look dark. Why it's called dark field illumination. And that's because any light coming directly from the lamp is blocked, and so it's not observed by the microscope. The only light coming through the specimen is light coming at such a strong angle that the light hitting the specimen, if it were not to be refracted by the specimen, that light would not be collected by the objective lens because that light is blocked by the opaque disc. However, some of the light that goes through the specimen is refracted away from its initial course, and it's refracted into a direction to be collected by the objective lens. And that's why this object looks darker than that object, because only the light rays that are refracted at a strong angle and refracted into the objective lens can be collected by the objective lens. All the light that is not refracted is blocked by the opaque disc, and it will never be collected by the objective lens. Any question about dark field illumination? So there's two things you should know, and that is we're looking at directly the light coming out of the lamp from the bright field illumination, and that's why the background of the object is very bright white, because all you're seeing is the light from the lamp. And then the specimen is brighter than the dark field specimen because we're getting more light coming from the lamp that's being collected by the objective lens. However, in dark field illumination, none of the light coming directly from the lamp is allowed to be collected by the objective lens, why the background looks dark, and then the specimen also looks darker because the only light being collected by the objective lens. It is light that is refracted by the specimen, and it has to be refracted at a fairly steep angle to shine it to the objective lens. Any question about the difference between bright field illumination and dark field illumination? We don't have any microscopes in the lab which use dark field illumination, but you do need to know these two types of illumination. The book talks about other types of microscopes. We're not even going to discuss them, so you don't need to know them. However, you do need to know about an electron microscope. Here we're looking at an electron microscope, and what do you notice about it being different than a light microscope? Shown, shown here. Anyone have a guess?
It's much larger.
Yeah, the electron microscope is much larger. An electron microscope does not use light to view the specimen. You use a beam of electrons to view the specimen. So you focus the electrons using an electromagnetic lens. You don't use a glass lens, you use an electromagnetic lens. And electrons have a much shorter wavelength than light. Trying to remember what a particle of light is called. A photon. It has much shorter wavelength than a photon. And that allows greater resolution so that we can magnify many times more with an electron microscope than you can with a light microscope. This allows for the examination of viruses, internal cell structures, and surfaces. There are two main types of electron microscopes. There's actually four types, but we're only going to talk mainly about two types. A transmission electron microscope, shown on the left here, and then a scanning electron microscope, shown on the right here. What do you notice? There are two things you can notice about what you're seeing. What's one difference between these two? The transmission electron microscope and the scanning electron microscope. Anyone?
That we can see within the cell body pretty clearly on the left.
Yeah, with the electron microscope, you can see within the cell. You're only seeing one plane, though, within the cell. And so to see the top of the cell, you'd have to uh, focus the electron microscope at a plane at the top of the cell, and the same at the bottom of the cell. So with the transmission electron microscope, you're seeing inside the cell, but you're seeing the specimen in only two dimensions, meaning one plane through the cell. With the scanning electron microscope, you are seeing the surface of the cell. You're not seeing inside the cell, you're seeing the surface of the cell, but you are seeing the cell in three dimensions. And those are the two main differences between the transmission electron microscope and the scanning electron microscope. Here we're looking at a scanning electron microscope. We're seeing the surface of this crystal in 3D. Here we're looking at a structure inside the cell. I think this is the chloroplast membrane of a cell. In its transmission electron microscope, you're seeing the specimen inside the cell, but only seeing two dimensions within the cell. There's a third type of a electron microscope. It's called the scanning tunneling microscope. You will not be examined on this, so you don't really need to know it. It uses a metal probe to scan a specimen. It does allow for the greatest resolution of an atom. The scanning tunneling microscope can see 1/100th the size of an atom. If you remember, the hydrogen atom is about the smallest, and we can see 1/100th the size of an atom. with the scanning tunneling microscope. There's also atomic force microscopy. You won't be examined on this fourth type of electron microscope. It uses a metal and a diamond probe inserted into the specimen. It does produce a three-dimensional image. Here is the toxin from Clostridium perfringens shown with an atomic force microscope. Any question about the electron microscopes? All right, let's talk about the cells you're seeing here. Which cells are easier to view? The cells on the left or the cells on the right?
Definitely the right.
Yeah, the cells on the right. The only difference between the cells on the left and the cells on the right is that the cells on the right have been stained with a dye. Like I said, you could call it stained by a stain. Dye and stain are used interchangeably. And the cells on the left are cells that are unstained. You can notice the cells on the left are very close to the contrast of the background of the slide, meaning the cell looks similar to the glass and maybe the water of the slide. Whereas the cell on the right has much higher contrast to the slide. And there might be water in the slide there too, because that looks like there's water there. And what the stain is doing is only increasing the contrast between the cell and the slide, the background of the slide. So with bright field light microscope, most microbes are colorless and they have little contrast with the slide or the medium that you put in the slide. It could be water. To see them, at least to see them well, you need to add a stain. Staining is coloring the specimen or the background of the slide to increase the visibility and increase the contrast between the specimen and the background of the slide, which for most cases, the background of the slide is just the glass of the slide. So if you want to look at cells, normally you prepare a bacterial smear, and let's put a thin film of solution of microbes on the slide. You need to make the bacterial smear prior to your staining. So a bacterial smear is a preparation you do before you do staining. When you do a bacterial smear, you don't have to stain it. You could look at the cells directly. And in fact, where do we have that? Right there on the slide here. We made a bacterial smear, but we did not stain them. So we're looking directly here at the bacterial smear. So a smear, my point is separate and a distinct procedure from the staining. And staining is a separate and distinct procedure for making the bacterial smear. Prior to do a stain, you normally make a bacterial smear. Do you ever make a stain without making a bacterial smear?
I don't think so. It would be
I
kind
don't
of
think
pointless,
so
wouldn't
either.
it?
Sorry, that wasn't a question. It was a question to myself. And
Oh,
that is,
okay.
I don't think you can. Um, I think whenever you stain a slide, you have to put it on a bacterial smear. But you do not have to stain a bacterial smear. So making a bacterial smear is putting the microbes on the smear. They're usually fixed onto the slide by air drying and then heat fixing them to attach the cells to the glass. A stain also called a dye, is a salt consisting of a positive or a negative ion. In a dye, one of the ions is colored. We call that the chromophore. A basic dye has the chromophore on the cation, the positive part of the salt molecule, meaning on the cation. The acidic dye is the chromophore on the anion, the negative part of the salt of the dye. Any question about any of that? So obviously we have two types of dyes, a basic dye and an acidic dye. Bacteria have a slightly negative charge. Therefore, basic dyes stain the cell better than an acidic dye. And that's because a basic dye is attracted to the slightly negative bacteria cell, whereas an acidic dye is actually repulsed from the bacteria cell. So basic dyes are used the most in microbiology, and then all of the common dyes that we've talked about, crystal violet, safranin, methylene blue. We'll talk about malachite green in just a little bit. All of these common dyes are basic dyes. Acidic dyes do not stain the cell. They do stain the background of the slide, meaning the glass of the slide, but they do not tend to stain the microbes. An example of an acidic dye is eosine, nigrosin, and acid fuchsin. We'll talk a little bit about acid fuchsin. I don't think we're going to actually talk about eosin and nigrosin, but we will briefly talk about acid fuchsin. Because an acidic dye stains the background of the cell and not the cell, we tend to call an acidic dye a negative staining. This has to do with the old Polaroid films, which you'd have a negative from the Polaroid film, and you could use the negative to make more colored slides or black and white slides, it doesn't matter. But the negative had the opposite, the luminosity of the specimen, meaning where the face was brightly colored, normally in the photograph, the negative would have the face darkly colored. So that's where the term negative staining comes from, the old Polaroid film, which I imagine most of you have never even seen a negative. Has anyone in class ever seen a old Polaroid negative?
Absolutely, but I'm an 80s baby, so a little older.
Okay, so you actually were around before the Polaroid films disappeared. Okay,
Absolutely.
all right. This is a simple stain using one dye. This is a basic dye, and this is a negative stain, meaning using an acidic dye. You can see that the basic stain is mainly staining the cells, and the acidic stain is mainly staining the background of the slide. However, you still can see the cells very nicely with an acidic dye. Any question about the difference? All right, there are three main types of stains in microbiology. There's a simple stain, there's differential stain, and then there are special stains. A simple stain is where you're staining the slide with a single dye. It can be an aqueous, or meaning a water-based or an alcohol-based dye, but you're only using one single dye, either acidic or basic, and it is a simple stain. The primary purpose of using a simple stain is simply to increase the contrast between the background of the slide and the specimen. and then you'll be able to see the cell better than if you don't use a stain. The stain is applied to a fixed smear for a certain length of time. The stain is then usually washed off. The slide is then air dried and examined. A simple stain may use a mordant. A mordant intensifies the stain of the specimen so that the staining sort of intensifies. Functions of a mordant are it is not a stain. Like in the Gram stain, we use a mordant. We'll talk about that in a minute. And it does not stain the cell, but it intensifies the stain, increasing the affinity between the stain and the specimen, like Gram's iodine increases the intensity of the crystal violet, which is the stain of the Gram stain. It can coat the specimen multiple times to enhance it, thereby making it easier to see by increasing the width of the object that you're viewing, just because you keep putting on layers and layers. The flagella stain, let me blow this slide up, shown here, has a mordant that you put on, and you put on many layers of mordant, and they all start staining the flagella, putting on layer and layer of mordant, so that the flagella become visible. Normally, under the light microscope with a stain, you would not be able to see the flagella. You have to put on the mordant, and then the mordant coats the flagella many times, and then you can start see the flagella. In this case, the flagellum would be that one. There's many of them on that one. Mini flagella. All right, any question between a simple stain and a not simple stain? To be a simple stain, you simply use one stain, and then all the cells will be colored the same. So you cannot distinguish, at least by the color, differences between cells with a simple stain. A differential stain is a stain that reacts differently with different kinds of microbes. Therefore, you can distinguish between two types of microbes. Usually, two dyes are required, and that makes the cells two different colors because you're using two different colors of stains, but you do not have to have two different dyes. Normally, you have two different dyes, but you do not have to have two different dyes. As long as the cells stain differently, you could only use one stain. Any question about a differential stain? The most commonly used differential stains are the Gram stain and the acid-fast stain. The Gram stain is a stain developed by Hans Christian Gram in 1884. You don't need to know the year that Hans Christian Gram discovered the Gram stain, but you should know that it was discovered by Dr. Gram. And so whenever you print out the Gram stain, you should capitalize the G because we capitalize somebody's last name. And the Gram stain is named after Dr. Gram. Any question about that? All right, Dr. Gram used the Gram stain to differentially stain a person's infection where the infected bacteria stain differently than the patient's own cells. And so Dr. Gram used it as a differential stain to separate the patient's cells, which stain pinkish, from the bacterial infection, which stain purplish. Microbiologists started using the Gram stain, and they soon learned that it was a very useful stain in bacteriology because it classified all bacteria into essentially two very large groups. the gram-positive cells, which stain a dark purple, and the gram-negative bacteria, which stain a pinkish reddish color. And you can separate almost all bacteria into either gram-positive or gram-negative cells. Any question about any of that? So that is why the gram stain is differential, although Hans Christian Gram intended it to be differential. He just didn't realize that it differentiated bacteria into two types of cells, gram-positive and gram-negative. Any question about any of that? All right, here's the brief outline of gram stain. You make a bacterial smear on a glass slide. This is not making the gram stain. It's just that when you're doing the gram stain, you have to apply it to a bacterial smear. So the bacterial smear is something you do, a procedure you do before you begin the procedure of the staining. So actually the procedure of the staining starts with step 3. All right, once you have the bacterial smear made, you then let it air dry, and then you gently heat fix the slide. That's to attach the bacterial cells to the glass of the slide. If you did not do the air drying and the heat fixing, then all the cells would wash off with the staining procedure and the cold water washes of the gram stain. The first thing you do to start the staining procedure is you then flood the smear, which has been air dried and heat fixed, with crystal violet. You put the crystal violet on the slide for 60 seconds or a minute. After a minute, you pour the gram stain off the slide and then gently wash the slide with cold water to remove the traces of the crystal violet. You then flood the slide with iodine. Iodine is a mordant. It does not color the cells directly, but it intensifies the staining of the crystal violet. And you apply the iodine for 60 seconds. You then dump the iodine off and then wash the slide with cold water once again. Take off the worst of the water, and then you flood the slide with the decolorizing agent, which is usually acetone ethanol, but there can be other decolorizing agents. And you apply the decolorizing agents for a brief period of time, only about 5 seconds. And we'll talk a little bit more about how long you do that in just a second in our next couple of slides. You then wash immediately with cold water. Why this step has to be immediately after step 7 is because when you pour off the decolorizing agent from the slide, there will be trace amounts of decolorizing agents in the cell. And those trace amounts of decolorizing agent will start removing the crystal violet iodine complex from the gram-positive cells if given enough time. So what you want to do is wash immediately to remove the decolorizing agent. And then you dump off the worst of the water and then flood the safranin, which is a counter stain, for 1 to 2 minutes. After that period of time, you then pour off the safranin and then wash the slide gently in cold water. Blot the slide dry, and then you can examine it under the microscope. All right, so this is the procedure of the gram stain and the bacterial smear beforehand. When you're discussing the procedure, you only need to state the direct steps: flood the side with crystal violet, and then after every step of the gram stain, you can say, and then after every step of the gram stain, wash the cell gently with cold water. You don't need to put each wash in there. You can if you want, it's not wrong. I'm just saying you just need to put it one place, either the beginning or the end. After every step of the gram stain, you wash the slide gently with cold water. Any question about any of that? All right, let's talk a little bit about the staining of cells using the gram stain. When you apply crystal violet, all of the cells, gram negative and gram positive, will be purple in color. And this color is a bluish purple color from the crystal violet. When you apply the iodine acts like a mordant. It will intensify the stain so that the staining will be a little more intense to the human eye. Oftentimes you can't detect it, but under the light microscope you could, especially under the light electron microscope you could. But it's a little more intense. It's also a little bit more purple color than blue purple. So if you want to call this blue purple and this purple purple, that's fine. However, you just say purple and purple, that's also fine. The important thing is the iodine doesn't color the cells, it just intensifies the color from the crystal violet. And what happens is the iodine will complex with the crystal violet. And the iodine crystal violet complex will be a larger molecule than the iodine or the crystal violet alone. And that's why the mordant works. It makes a larger compound, intensifying the stain of the gram stain. We then apply the decolorization step, and that will wash the crystal violet iodine complex out of the gram negative cells, but not out of the gram positive cells. So the decolorizing step is actually the critical step of the gram stain, because at this point the cells will look different. The gram negative cells will be clear, and then the gram positive cells will be purple. It's hard to see the clear cells, so then we apply safranin. Safranin is a counter stain. A counter stain just means it's the second stain we're using after crystal violet, and it's a different color than the crystal violet. Why it's called a counter stain. And then that will stain the gram negative cells pinkish by the safranin. Any questions about the coloration of the cells at the different steps of the gram stain? And then the only other thing you need to know is it's the decolorization step, which is the differential step of the gram stain, where the two cells now look different. All right, any question about any of that? So let's go through the gram stain procedure in a little more detail. The first thing you do in the staining procedure is step 3, flooding the slide with crystal violet. It's a stain, it's actually a basic stain. You apply for 1 minute. The crystal violet sticks to the peptidoglycan in the bacterial cell wall. It is considered the primary stain because it's the first stain we're using, and it colors all the cells the same color, purple. After that, you then dump the crystal violet off and then wash the cell with cold water. The color of the gram positive cells are purple. The color of the gram negative cells are purple. You then dump the worst of the water off, and then you apply iodine. Iodine is a mordant. It increases the affinity of the crystal violet for the specimen because the iodine complexes with the crystal violet making a larger molecule than either iodine or crystal violet alone. You apply the iodine for about a minute, 60 seconds, and then the color of the gram-positive and the gram-negative cells is the same, a purple color. It is a little bit more of a purple color than the blue-purple of the crystal violet alone. But other than that, the two cells look the same. The next step is the decolorizing agent. We're going to talk about it here as the acetone ethanol decolorizing agent, but there are other decolorizing agents. There's acid ethanol, which works very similar to acetone ethanol decolorizing agent, and then there's simply 95% ethanol, which can be used with the decolorizing agent. The point of the decolorizing agent is to wash the crystal violet out of the gram-negative cells, but not out of the gram-positive cells. As long as you stop the decolorization and do not overwash, the gram-positive cells will retain the crystal violet iodine complex. The decolorizing step is considered the differential step of the gram stain because at this step, the gram-negative cells and the gram-positive cells look different. They stain different. How you apply the decolorizing agent, the best way, pretend this is a slide, hopefully you can see it against my face. Get the decolorizing agent, put it up to your eye, and add the decolorizing agent on the top of the slide, holding the slide at an angle, running the decolorizing agent down the slide. When the crystal violet iodine complex stops running, when you stop seeing purple running down the slide, you know that you've applied the decolorizing agent long enough, so you should stop applying the decolorizing agent. The reason being is that if you were to put too much decolorizing agent on for too much time, you will start to decolorize the gram-positive cells, and we don't want that to happen. You then wash immediately with cold water to remove the trace molecules of decolorizing agent, which for here we're talking about the acetone ethanol. The color of the cells, the gram-positive cells will stain purple color, and the gram-negative cells will stain colorless, at least if you've applied the decolorizing agent for the right amount of time. If you don't, you could pull the purple out of the gram-positive cells, meaning you over-decolorize, but you can also under-decolorize, where you fail to pull out the crystal violet iodine complex from the gram-negative cells, in which case they will stain purplish in color. Any question about any of that? I guess what I'm trying to say is the decolorizing agent and the decolorizing step are the tricky ones. You have to apply the decolorizing agent for the correct amount of time. Too long, and you pull out the crystal violet iodine complex from the gram-positive cells. Not long enough, and you do not pull out all of the crystal violet iodine complex from the gram-negative cells. And then you will have both cells staining the same. So it's not a differential step. All right, safranin is a pink dye. It does stain all bacteria. We use it as a counter stain because it has a different color than the primary stain, which is crystal violet. You then flood the safranin for about a minute on the slide, and then after the minute, you dump off the safranin and wash the slide gently with cold water. And so in my procedure, if you simply apply all the agents except for the decolorizing agent for 1 minute, you now know the procedure of the gram stain. And then just you state something like you apply the decolorizing agent until the color stops streaking on the slide, but you could also say for about 5 seconds. Although the safranin is pink in color and stains all bacteria, after the counter stain is applied, we will see that the gram-negative cells stain pink, but the gram-positive cells stain purple. Why is it that the gram-positive cells stain purple if I just told you that the safranin stains all the bacteria, not just the gram-negative cells? Anyone have a guess?
Is it because the original dye is darker?
Say again, I didn't quite hear you.
Is it because like the original dye is darker, so even if you dye it with the safranin, it kind of like overshadows that?
Yes, with the human eye, all we can see is the purple in the gram-positive cells because the purple is more intense of a color than the pink, and our human eye can only see the purple. Even with the light microscope, you can only see the purple. But the pink is there. If you look at the cell under the electron microscope, you would see some parts of the gram-positive cells staining purple and other parts of the gram-positive cell staining pink. So you can see the safranin in the gram-positive cells if you're using the electron microscope. But the human eye cannot see the pink. It only sees the purple. Any question about any of that? You then blot the slide with bibulus paper. In case I haven't told you, bibulus paper is thick paper, sort of like thin cardboard. We're going to pretend this is bibulus paper, and you just rip it out of the book. Now, this is just regular paper, but rip it out of the book. And then what you do is you fold it over. I'm just going to pretend that's the fold. Can you guys see this? Anyone? I'm going to assume you can see this. Put the slide in the bibulus fold and then blot the bibulus paper down onto the slide. And that will remove enough of the water that you can then view the slide, even with oil, under the light microscope. Meaning there will be trace amounts of water left behind, but you will be able to remove the majority of the water so you can even use oil on the slide. When you're using the bibulus paper, you make sure you do not rub the bibulus paper this way or that way. And also you make sure you, well, I guess the same. You make sure you don't move the slide in the bibulus paper. You just blot it dry. If you were to rub it, you're going to rub off the cells. If you just blot it dry, you do not remove the majority of the cells. I'm not saying you don't remove any cells, but you don't remove the majority of the cells. Any question about any of that? All right. We'll stop here talking about the basis of the gram staining next time. Any questions?
I just have one request. Is it possible that you post the slides for each chapter before the quizzes? Because I didn't see the chapter 2 slides before the quiz yesterday.
That's because we hadn't finished chapter 2.
Gotcha. So you won't post the slides until we've already gone through all the slides together?
Yes, it is a problem. Did we have the quiz on chapter 2? If you let me know in advance, I will try and post the slides beforehand. I used to have that, but I don't have an earlier one, but I can make another one and make a link to it. Um, but I do not post the notes until after the chapter is done because I really want to encourage you to take notes yourself. I don't want you to use my notes. Just that you will not learn as well as you if you use only my notes. So before the quiz next Wednesday, if I don't have chapter 2 posted, just send me a reminder. We should finish chapter 2 easily by next Tuesday. I think we're supposed to finish it by today, but let me take a look here. Yeah, we were supposed to finish it by today, but we didn't get started. We were actually supposed to start chapter 3 today. We're still on chapter 2. So I'm a little behind schedule. All right. Any other questions? If not, I'll
Will
see
we,
you in the lab.
sorry, will we have a chance to review our last quiz at all just to figure out where we went wrong, or is there any opportunity for that?
Yeah, you can see answers to your quiz. Um, I believe Saturday morning. It might be Saturday evening, but the answers will show correctly Saturday. I make a 2-day delay because some people uh, request to take the quiz late, and I always tell them it has to be taken no later than 2 days because after 2 days the answers will show, and if once the answers show, you can no longer take the quiz.
Understood. Thank you so
All
much.
right.